Good afternoon, everybody, and welcome to our next SAEON seminar. So today we have Helga, who is the biodiversity landscape scientist at EFTEON. Well, the landscape scientist for biodiversity. I mixed them around. Sorry about that. And she's going to be chatting to us about a little bit of a work she did at Knoetze during a postdoc there. So she's had some experience in some different aspects of SAEON. So we're excited to hear more about your talk today. So thank you, Helga.
Hi, thanks, Caitlin. Hello, everyone. Greeting you all the way from Kimberley. And I'd just like to thank you for joining the seminar where I'll be talking about megacarcasses and how they are a unique disturbance type and how they add to the spatial complexity of semi-arid savannas. Now, like Caitlin said, this is results that formed part of my postdoc study under the mentorship of Prof. Dave there at Knoetze. And we are a bunch of international and multidisciplinary scientists from America, Norway, South Africa, and I think Johan is currently in London. So we are botanists, soil scientists, large mammal ecologists. Who did I miss? Entomologists and also a social scientist that are trying to tease out the ecological legacy effects of megacarcasses in African savanna ecosystems. And we also have a paper that we published recently, so please go and have a look at that. And I hope this is going to be a jaw-dropping experience for you. So we know that decomposing large vertebrate carcasses are a natural disturbance type. They are a localized source of nutrient enrichment, and they have strong implications on ecosystem biogeochemistry and also the community dynamics of just about everything: the large mammals, carnivores, vascular plants, the soil, and then also the soil microbes. But we have limited knowledge on how individual carcasses affect the spatiotemporal heterogeneity in terrestrial ecosystems. Surprisingly, we have a much clearer understanding of what happens in marine ecosystems, where the megacarcasses of whales create these oases of biodiversity and productivity that can actually last for years. But despite being the largest land animal, we know little about how the nutrients from the megacarcasses of the African elephant affect savanna ecosystems. And we know that elephant mortality is a natural part of the savannas where they still occur. And for example, in the Kruger National Park, annual mortality can total several hundred individuals annually. So nutrient pulses of about 800 kilograms of carbon, 300 kilograms of nitrogen, and 125 kilograms of phosphorus for a single 4-ton individual, and their impacts are likely to operate at a landscape scale. So these nutrient pulses then create nutrient hotspots, and these hotspots can have long-lasting legacy effects on the ecosystem and also the processes occurring there. And it's also known that these carcass-derived nutrients causes shifts in plant communities. So we anticipated that carcass sites will be dominated by unique sets of species and also traits, and that these species will likely be adapted to high fertility soil, or the species occurring there would have traits for them to be adapted to high fertility soil. So the data that I'm presenting today is data that we collected on the megacarcasses of 10 elephants that we sampled on granitic and basaltic soil in the Kruger National Park. So at each carcass site, we established 4 transects of 15 meters in length in each of the 4 cardinal directions. And we sampled vegetation within 1 meter squared subplots and we placed those plots at various distances away from the carcass. As you can see, at 1 meter, 2.5, 5, 10, and 14 meters away from the carcass. And within these 1 meter squared subplots, we identified all the life forms, and we also noted the total percentage cover of vegetation, bare soil, bone, rock, litter, and dung. We considered the carcass sites to be 0 to 3.5 meters away from the carcass. This was based on field observations that we made while we were visiting the carcasses, where you could really clearly see a zone of impact of these carcasses. And then the matrix sites were considered to be 10 meters and further away. And these field-based observations were then further corroborated by soil nutrient analyses that we did. And I'd just like to thank Courtney for creating these graphs. And as you can see that soil ammonium and soil nitrate, as well as soil phosphorus, was much higher closer to the carcass or at the carcass sites compared to the matrix sites that were further away. And Courtney also acknowledged the age of the carcass, but you can see that there was a similar trend irrespective of how old the carcass was. And then similarly, one of the dominant species that we found was Erochlua trichopus, formerly known as Erochlua mosambicensis. And we also found that the foliar nitrogen content of this species declined as you moved further away from the carcass. So these levels of foliar nitrogen was also much higher at the carcass sites. So what did we find? We found that the vegetation communities from carcass and matrix sites were floristically distinct. And this was probably due to the abrupt changes in the soil nutrient concentrations, as I just showed, the higher levels of nitrogen, ammonium, and phosphorus. And this input of nutrients into the soil causes what literature calls cadaver decomposition islands. So as you can see in this photo, it is these nutrient-rich bare patches that actually facilitate the recruitment of species that would otherwise not have established there. And it also supports vigorous plant growth during secondary succession. So yes, we found that, interestingly, that the species that contributed most to the significant differences in species composition between carcass and matrix sites were mainly grasses. So the higher levels of soil nitrogen and phosphorus at the carcass sites likely causes the formation of biogeochemical hotspots, and we know that these hotspots are predisposed to form grazing lawns. And inherently, these grazing lawns attract increased herbivore activity, and then this in turn helps maintain a high productivity in grasses. But it's just important to remember that this was a snapshot in time, and probably if we were to isolate carcasses with more early succession or even mid-succession and late succession, these response patterns might be totally different. So if we zoom in a little more on what happened at the carcass sites and the dominant species occurring there, it was interesting to find that all the species that was dominant at carcass sites were palatable species, or species that had a higher quality of forage. And this is to be expected on nutrient hotspots. 80% of these species were perennial grasses, which was interesting for us because we would have expected more annual species because of the higher intensity grazing and also inherently the higher disturbance levels close to the carcass sites. And then, although we know that forbs or the wildflowers, they tend to perform better with increased soil conditions, but grasses tended to dominate these sites. And then on a functional trait level, it was interesting to see that 60% of the species exhibited a prostrate growth form. So that is when the species grows very close to the soil surface and in a flat way, which is then also a functional trait which species, an adaptation that allows species spatial refuge from grazing. And these species generally are also nutritious and potentially high bulk density forage resources for the animals in that area. Considering the matrix sites and the species that dominated there, it was also, it was interestingly mostly perennial tufted grasses. And this, for a person that loves forbs very much, was a bit of a bummer. But yeah, like I said, perhaps the patterns will be different if we isolate the different ages of the carcasses. But yes, we found mostly perennial tufted grasses, and the lack of a high forb abundance could be attributed to these tall, fast-growing tufted grasses that outcompete forbs of a lower stature for resources such as light, water, and nutrients. And then the occurrence of these grasses, the high levels of productivity, and then also the lower levels of grazing, perhaps a bit further away from the carcass site, then favors species with traits such as tufted graminoids with a high leaf production. And then interestingly, most of these species found in the matrix sites, in other words, a bit further away from the carcass, about 71% were also palatable species, which just reinforces the idea that these carcasses become zones of fertility and favors the establishment of high quality forage for animals. So, yeah, this study contributes to a growing recognition that these megacarcasses do have an effect on terrestrial ecosystems, and the effect of elephant carcasses on the herbaceous layer, the structure and function of herbaceous layers that we demonstrate here for the first time expands this recognition significantly, given that elephant mortality is a natural phenomenon in the Kruger National Park. And importantly, if we lose these mega herbivores from the ecosystem, we also lose these potential nutrient hotspots to occur within the landscape as well. Yes, so megacarcasses therefore represent a unique disturbance type, and they add to the spatial complexity of semi-arid savannas by creating these nutrient hotspots, and these hotspots have distinct floristics and increased forage potential. So what should you take home from this today? Elephant carcasses appear to play an important role in terrestrial ecosystems of semi-arid savannas. They influence plant community composition and structure. As we saw, the carcass sites were floristically and structurally distinct from the surrounding matrix. And interestingly, the floristic differences in this study was mainly driven by grasses, but we do need some more long-term studies to explore how the other life forms would respond to these carcasses. It's always important to remember the time component. So we would also expect these response patterns to change with time. And then megacarcass sites are considered nutrient hotspots, supporting mainly palatable species, and they are therefore considered functionally important for semi-arid savanna landscapes. So what's next for me, at least, in terms of this project? I'm currently looking at the role that these megacarcasses play in driving local patterns in plant diversity and richness, and I'll be presenting some of these results at GSSA in July. And then also on the planning for the year is to see how these megacarcasses affect the decomposition rate of organic material. And for this, I'm going to use some tea bag index data that I collected in and around the carcass sites, and I want to link that with some soil respiration data. Just want to thank Nate for compiling these graphs so long, just to show you that the CO2 respiration was much higher closer to the carcasses, if this is the distance closest to further away from the carcass, we could see, at least also for the granite and the granitic sites, that the respiration rate was much higher closer to the carcass compared to further away. So really excited to have a look at how those interactions work. And with that, I would like to thank you for your attention. And yes, Caitlin is going to assist if there are any questions.
Thank you, Helga, for a nice, short and sweet seminar. Yeah, I think it's quite cool because we always think about how elephants like affect trees and stuff, but you don't think about what happens when they die and how they affect the herbaceous stuff. So I want to now open it up to questions and see if anyone has questions, and hopefully we can have some nice chats about it. Would you like to ask your question?
Thank you, Caitlin. Hello, Helga. I hope you can hear me.
Yes, I can. Hello, Nannies.
Okay, cool. Helga, I am interested to know, why did you choose to work on the granites and the basalts? Is there a specific reason? Because I'm quite interested to know how would the control plots compare to your fertile island, or how the two differ from one another. So that I'm just curious.
Thank you, Nanette. And can I just say she's asking the question all the way from Japan. She's in Japan currently for a conference. So I feel so special that she joined at late at night. Nani, so the whole soil dynamics is also one of the major hypotheses of the bigger project. That's why we asked the rangers to let us know about carcasses on both of the soil types, especially since it's the main soil types of the Kruger Park. So, and we know that the granites are, that's the nutrient poor one, and the basalts are more nutrient rich. So we would expect dynamic interactions, and I think the soil scientists are really interested in seeing how that's going to pan out. So yeah, that's how they decided at the end to sample carcasses on both of these major soil types in the Kruger Park.
Cool, thank you. I'm looking forward to see your next results from the study. It's very interesting, and your presentation was fantastic.
Thank you, Nanis.
Kathleen, I see your hands up. Would you like to ask your question?
Thanks, Caitlin. Thanks, Helga. So my question, I'm going to ask it. It's a little bit vague, but I suppose I'm asking. So you targeted elephants obviously because of their body size, because it's such a big pulse. Are you considering looking at smaller mammals? I mean, did you pick the elephant because of its quantity, or do you think that there would be anything different in the ratios that they would pump into the system? In other words, I suppose I'm asking, these hotspots must occur for all carcasses. I wonder if there's a functional change at size, or maybe whether they're a grazer or a browser or predator. Has this been discussed in your group at all?
Thanks, Kathy. Yeah, very interesting question. And it is something that I've been having in the back of my mind to see if mesoherbivores, of course, they will also have an effect on the ecosystem, but I think we chose the elephant because maybe of the longer lasting effects that they might have because of their sheer size. We would expect that these effects would remain in the system for a longer time period. But yes, of course, I have been thinking about Benfontein and the smaller antelope species occurring there. And of course, they won't have such a big impact in the landscape, but I am still interested to maybe compare that at one stage or another. I don't think the group is discussing that at this stage. They are actually building on this to even elaborate to other countries such as Botswana and Zimbabwe. But yes, a very interesting thing to keep in mind that I, at least in my capacity, would like to explore some more.
Well, thank you for that. Tsimbetso, would you like to ask your question?
Okay. Thanks, Helga, for the nice talk. You mentioned that the loss of these elephants or mega herbivores could disrupt nutrient cycling and reduce the forage quality. Two questions in one. How will this knowledge influence conservation strategies or rangeland management? And could maybe targeted carcass placement or alternative nutrient supplement mimic these disturbances, maybe just to maintain this ecosystem functionality?
Hi, Timur. Thank you for the interesting question. So firstly, I think this research is very important to show, like Caitlin said, we know that when elephants are alive, they are ecosystem engineers and they shape the environment through breaking the trees and eating a lot of the vegetation. But I think it is really important for management to see that they actually have big and long-term effects even after they die. So yes, I think it's really important to share this with management, and they can see that if you were to lose these big herbivores from the system, you're also going to lose the potential of having these biogeochemical hotspots in the landscape. And then if I understand the second question correctly, I think it's like Kath also asked, I think it's totally possible to try and mimic the effects of these animals if they were to be lost by using other herbivores to still be able to maintain that function in the ecosystem of developing these nutrient hotspots and then the grazing lawns, and then this can be a long-term effect and a long-term grazing lawn for herbivores to use. So yeah, I guess it is a possibility.
Well, thank you. Do we have any other questions that people would like to ask? Tsimbetso, you got another one.
No, it's just a quick one. So, like you, I was surprised why were the perennials maybe dominating in those centers. But then maybe the question is, I'm not sure if maybe you did look at, were they coming from maybe seeds or way like maybe dormant buds that were just there waiting for this nutrient deposition for them to regrow. I think that could be quite maybe a good mini study to check if they were coming from the seeds or from the buds.
Yes, and you are the bud specialist, especially in terms of Thorpe Tumbe. So yes, that's most probably another research question that we can literally dig into. I have no idea if it was from seed or if it was just the bud bank that was just ready to grow. I have no idea, but it's a cool question, and it's something important to consider for some future studies, maybe.
Well, thank you. Yeah, Tetsi. Sorry, I might have got your name wrong, but I see there's another hand. Would you like to ask your question?
Hi, Helga. Hi, Kathleen.
Hello,
Sorry.
Tim.
I didn't realize I didn't unmute myself. I just wanted to ask, like, with the elephants, right? So you sort of like on their carcasses, you sort of like have vultures and other scavengers coming in. And I know like you have like almost from another study that was done in Zimbabwe, almost have like 200 vultures coming into those carcasses. So probably they also defecate and leave a lot of excreta there. Do you consider that in the study, just out of curiosity?
Yeah, so Titi, thank you. Yeah, that's an interesting question. We do have camera traps that we put up at these carcass sites, and it's some of my colleagues are looking at the behavior of the animals, be it the vultures or the scavengers or even other elephants that come and visit. But I don't think that was one of the original things that we considered actually. But yeah, I mean, it can definitely help in explaining these higher levels of nutrients closer to compared to further away. Yeah, no, that's another interesting perspective to keep in mind, perhaps to help us discuss what we see. Yeah, thanks for that, Titi.
Cool, thank you. And I think it's quite nice, these discussions, to start thinking about, you get ideas of other things to start thinking about, which is quite nice.
Yeah.
Does anybody else have a question they'd like to ask or anything they'd want to discuss?
Because I always say the more that we find out about something, the more we realize that we don't know a lot about it. And then there's just a lot of other questions that pop up and a lot of things that we can still research. Yeah.
It's nice to get people who have had slightly different backgrounds, their thoughts on stuff.
Yeah.
Lucian, I think you just unmuted, Bob. Okay, would
Yes.
you like to ask your question?
Yes, I'm trying to find the hand. I can't find the hand. Helga, thanks for your talk, man. Maybe just a comment from my side. If you're looking at the nutrients and so on, maybe it would be good to add the bacterial component to your study. You can do great things now with the microbiome type stuff. There's certain bacteria that will break down nitrogen or phosphorus, or you can get signals or hits, you know, to help build your picture better. So just a comment from my side.
Thank you so much, Lucian. I made a note. Thank you so much for that.
Cool, cool.
Well, thank you, Lucian. Is there anybody else who'd like to ask a question or suggestion? Yeah, we all happy. No more burning questions. Perfect. Thank you so much, Helga, for a very interesting talk. I definitely learned a lot, and it made me think about, you don't have to just think about what things do when they're alive, also what happens there, like legacies after they die. And I think it's quite nice to start thinking about things as like a whole. We don't always do that too well. So yeah, thank you, Helga, for an interesting talk, and thank you everybody for joining today. I want to invite you to our next seminar next month, so it will be on the 3rd of July. We're still confirming the speaker, but we hope to see you there, and this recording will also be up on the SAEON YouTube page, hopefully today, but definitely by tomorrow. So thank you so much for joining us, and hope to see you next month.
Thanks, Caitlin. Bye, everyone. Thanks for
Bye,
joining.
everyone.
Thank
Thank
you.
you.
Bye.
Bye.
Yeah.